All right, so one last thing, just to make sure it's clear. So DNA versus RNA. What's going on? So I don't need to draw any more complex molecules. I found on all my drawings, so we're back to writing as we go. So DNA, deoxyribonucleic acid. DNA. This is long-term genetic information storage. So you don't want anything happening to it. And I keep referring to it as kind of like our master copy. Okay, this is the copy. If anything happens to this copy, it's officially it's a different set of DNA. Like the organism now has a change. Like there's no going back. So we want to avoid that for the most part. We want our genetic diversity to come from things like reproduction, where we're mixing and matching DNA through reproductive processes. You don't want it to come from constant radiation. Now it is a research technique. Something that's been done with plants. They would just irradiate seeds and see what happened. So I mean, it's not like it didn't happen, but it's not a preferred method, through natural selection. That's not generally how natural selection works. Generally, DNA of an organism isn't changing, but the diversity of the offspring is kind of helping select, or the environment is helping select what is going to be beneficial in offspring versus not, and those offspring's ability to reproduce themselves is going to change the genetic makeup of the population to match the needs of the environment. So it wasn't like a giraffe got hit by a comet and then all of a sudden its neck went whoop. It happened over many, many, many generations. That neck just got longer and longer and longer, until we get to what we see today with giraffes, which are misshapen horses. If you don't believe me, watch them run. And I don't know how a giraffe exists. Some things in this world just make no sense. All right, RNA. Someone out there is going to be a giraffe apologist and jump on me for this. I don't want to hear it. All right, the giraffe lobby will not sway me. We've got ribonucleic acid. All right, and so RNA. And this is kind of like our cheap copy. So this is short-term genetic information. So the DNA is it's stored in the cell's nucleus. It's like in a vault. You think, well, it's in a vault. It's a master copy in a vault. You can look at it and you can make a copy, but you cannot take the original copy out with you. You can only take the photocopy out with you. RNA is that photocopy. So this is, that's long term. We'll say this is short term because it does degrade in the cell. But it leaves the nucleus. To be used for protein production. And you can use the same strand of RNA over and over and over again as long as it doesn't degrade. So it's not like you make a copy, you usually get one protein. So it's not like the DNA is that active. You send out the RNA, the cell hangs on to it, and when it needs it, boom, starts making that protein. There's all sorts of ways of triggering those processes depending on the protein. Lots of factors can go incredibly complicated. Like I said, I used to be a biochemist. I don't regret switching tracks to physical chemistry. I'd much rather do quantum mechanics than think about the cascade effects of everything. All right, so it leaves nucleus to be used for protein production. So this is the cheap copy. This is what you hand. This is what you hand the guy like me on the ground. I've got to dig the holes to pour the foundation footings or whatever. I got to know where to do it. What do I do? I look at my blueprint. Is my blueprint the master copy? Oh, there's a problem in the world if I have the master copy in my hand when I'm the guy that digs holes in the ground. Okay, so I get the cheap copy. All right. If I accidentally leave it in one of those holes or spill coffee on it or whatever, I've seen so many things happen to blueprints over the years, or just flat out lose them. They just get lost. They blow away in the wind. You leave them in the truck, and then something's put on top of them. You can't find them. These things are real life issues I've come across. You just get another copy. If you lose the master, you don't know how to make a building anymore. If you lose a copy, you just get yelled at, and then they give you another photocopy. Which is much more preferential to just not knowing how to make the building. So RNA is the cheap copy that gets sent to the people on the ground. The people on the ground level, they're doing the work. They got to know what they're doing. They've got to have plans, but they don't need the original. They can just get a copy. All right, so the differences. So I've talked about the structural differences, and now I've hopefully cleared up the functional differences as well. They're in different parts of the cell. RNA travels around. If you have DNA floating around freely in the cell, that's one messed up cell. Okay, it needs to be locked down in a vault. It's going to be wrapped around histones and the chromosomes and very, very tightly controlled. So, when you're looking at DNA versus RNA, there's difference in their functions, and then there is a couple other differences. So you have like the AU base pairing. I spent way too much time in these drawings, just throw them away. This is going to be an RNA versus the AT that you'll see in DNA. Now GC pairing, that'll occur in both. So DNA or RNA, you see G or C, whatever. You see A, you don't know. If you see T, you see a thymine in your chain, you know you got DNA. If you see a uracil, you know you got RNA. But a much easier way to tell them apart is the part where it's the difference in the name, and that's just what sugar are you looking at. You see an OH group here, it's ribose, it's RNA. You see no OH group on this carbon. You just see two H's, it's deoxyribose. You've got DNA. It's as simple as that. So, and they'll both have phosphates. So they both have sugar phosphate backbones. They just have different sugars, and then there's that one base that can be different between them. But it's a lot easier to look for the sugar difference. And so that's really what you want to do to tell them apart. And hopefully that kind of clears up what the difference is between DNA and RNA. So there's different types of RNA. There's like tRNA and mRNA. And I'm going to let your biology instructor talk about that. But there's still RNA. All that little letter in the front is telling you is what that RNA gets used for. Is it used for transcription? Is it used for messaging? Whatever it is. So there's a few other kinds too. And there are even, sometimes RNA can play a bigger role in certain organisms or in viruses as well. But I'm not going to get into that. I'm mostly sticking to what's going on in eukaryotic cells, our cells, animal cells. All right, so I will leave it there, and this will be the end of this section. So again, every biomolecule we talk about, we're going to go small, big, 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 big. So we've taken all the way to the macromolecule now. Oh, actually, DNA, double-stranded, right? 2 strands. I talked about this earlier. RNA, single-stranded. So that's the other difference too. It's a cheap copy. We don't need to protect the bases on it. If something happens to them, we just send out a new copy. DNA, double-stranded, helps increase data fidelity, and also is very important for cell replication. Because this strand so we split them, we duplicate them, and then we have two new sets. All right, so yeah, I'll leave it there. Those are the differences. And that'll do it for this section in terms of lecture videos. And I think everything's there to kind of help you through the homework and the concept check and everything. A lot of this stuff I talked about, I tried to be clear, like you don't need to know the structures, you don't need to know that much of what I talked about, honestly, for this class, but I really wanted to help you out with the biology. So a lot of it was kind of over the top for the chemistry side, but that was just to help kind of soften the blow. This is heavy duty stuff on the biology side. And I just wanted to do my part to help you out there. So hopefully you did. If not, you can ask your biology instructor lots of questions. All right.